All right, Jason Curry, Bio 225, Week 7 results of Exercise 1, the Kirby-Bauer diffusion test, and Exercise 2, investigating penicillium. All right, so my area was wiped down with bleach. All my products are sterile with bleach, and I have my gloves on, and we're ready to start the exam. So this is a result after incubating about 12 hours. There was a positive change to this. The novobiocin seemed to show the most, I guess, would you say, susceptible to the S. epidermis. The penicillin also had a susceptibility to the epididymis, and the gentamicin also showed, but it had the least, so it was a little more resistant to the antibiotics. My hypothesis, I did think the novobiocin was going to have the best results just based on the study. It showed it ruptured DNA cells, and it proved to be correct. The penicillin I did not think was going to work as well because of the way the S. epididymis responds and mutates and changes. And I thought just because of the overexposure of penicillin has mutated this bacteria. And then the gentamicin was more of a full spectrum antibiotic, so I didn't think that was going to be strong enough based on the resistance of the S. epididymis and how it responds well to antibiotics in a way where it inhibits growth instead of destroys growth. So what I'm going to do is I'm going to take measurements to see how this came about. So let's do the gentamicin first. I'm going to measure in millimeters, and that was about 2.5 millimeters. So that's 2.5 centimeters, so that's about 25 millimeters. Let's do the novobiocin. That is about 35 millimeters. And then the penicillin, measures up to about 30 millimeters. So the novobiocin had the largest response. The penicillin came into second and had the next, was more susceptible. And then the gentamicin seemed to be more resistant to the S. epididymis. So that was that result for lab 1. So let's move on to exercise 2. Still see my name there. All right, so exercise 2 is investigating the penicillium. Penicillium is naturally found in the environment. It is gram stain positive. It prevents bacterial growth and will explode cells once it's exposed to the bacteria. So it's a real useful antibiotic for the general population. For me, I'm actually allergic to it, so I was kind of iffy with this. When you look at the slide, let's turn it on. I don't know if I'm going to be able to show you the slide here. I can't flip my camera while I'm doing this. I'll try to line it up on this way, but I don't know how that's going to match up. It doesn't look like I'm able to do it, so I don't really know how to show you that. But I did take a picture, and it is in the HOL site with the picture. What we're seeing is reproductive structures of the penicillium. There's the conidia. It's an asexual spore, and they are grouped together externally once they are fully matured. The condydium is basically just a single asexual pore, and then phyllidide are single cell canindas. So it was really interesting to see the structure of the penicillium, and that is the results of my experiments. Thank you.